然后第4个方面就涉及到这个污染物的就是修复方面的，就是污染物控制方面的技术，就是大概就这4个方向。
好的。
然后现在就是说，我们已经出了一期的，就是1年了吧，2002年我们正式出刊了，反正就是大概有1年的这种这个出版。现在主要可能就是就是在推广这些方面，就是希望我们张老师能在国外的话，就是多多关注，或者是Twitter方面就是进行一些宣传啊。
好的。
谢谢张老师。
它这个旗舰期刊也欢迎引用里面的文章，因为现在呢，这个因为你作为一个旗舰期刊，你肯定这个引用次数要上去嘛，尤其是在国外引用，从美国引用，这个能提高期刊的影响力，
是
因为今天可能有点早了，再一个呢，出现就是周末以后我假如安排这个会议的话，尽量安排在工作日，我估计好一点点。
不是那个，就是何老师和张老师，我们现在还有2个，一个是我们的哔哩哔哩网站，因为现在很多国内的话，它是就是上这个 Zoom 不是太方便，然后他们是通过那个 B 站，还有我们那个有构想，然后可能周末吧我们是通过这2个平台直播的，所以我们那个张老师可能 YouTube 也是在直播的，所以您到时候可以
YouTube 直播，
到时候反正这个，因为它Zoom平台上有时候国内听的是不是太方便，可能是这样的。
行，好。
对，这个哔哩哔哩的网站上还是挺多的。
我看了一下。
Okay, now, so it's 9 o'clock. So I would like to express that I'm very glad to invite Dr. Ke Zhang to be the speaker in our platform from EEH. So now, give me 3 minutes to do a brief introduction about Dr. Zhang. Dr. Zhang is a postdoc researcher in the Department of Civil and Environmental Engineering at the University of Michigan. He received a bachelor's degree in Zhengzhou University and a master's degree in University of Chinese Academy of Science. He earned his PhD degree in Energy, Environmental Chemical Engineering from Washington University in St. Louis, under the guidance of Kimberly, is right? So
yeah.
He had a unique research background in both microbiology and environment engineering, which is a very integrative field. His research interest is to study environmental biochemistry to address environmental challenges from nature and engineered biological pollutants. His ultimate research goal is to predict the behavior of biopollutants in environmental system and to optimize strategies to minimize the risk proposed by these biopollutants to humans and eco-health. His current research is study the transmission of flu virus indoor environment, which is a very hot topic from COVID-19. Specifically, he collaborates with virologists to study the decay of flu virus on the surface. He also developed methods to efficiently detect flu virus and assess their effectiveness. During his PhD, he studies the environmental fate of RNA interface biopesticides, which are relevant to genetically modified crops. Her talk today is mainly about his findings during his PhD in St. Louis. He is a productive researcher, has several journal publications, including 3 leading authored papers published in the SI journal during his master degrees, and 3 leading author papers published in EST. Is very attractive and very good journal in the energy environment engineering. He also received several awards, including the 2 Traveler Awards from ASA conferences. Now, let's welcome his talk.
Thank you for introduction, Dr. So I can share my screen now. Okay, so can you see my screen now?
Yes, perfect.
Okay, good. Yeah, so today my topic is the environmental fate of ion interference by pesticide. I know there are many senior scientists have given talks in this platform. I'm just a junior scientist. So today it's just a glance at my field, biorisk assessment. We environmental scientists are familiar with potential hazards from chemicals to human and ecology. But potential hazards can not only from chemical, they can also come from organisms. For example, you should ever heard of some dangerous pathogen virus or microbes. For example, such as the SARS-CoV-2, Ebola, monkeypox. I currently study the environmental fate of flu virus in indoor environment. But I just started to study the flu virus from last year. So I'm not ready yet to share my findings on flu virus. Probably in the future, I can share my finding on flu virus on this platform or in other conferences. Biorisk can also come from engineered organisms. So if you work with engineered organisms at university, you may know that if you work with them, you cannot just treat engineered microbes as landfill garbage. You need to treat them as biowaste, which need special treatment. This is because they may bring potential hazards and potential unexpected hazard to the environment. Since I have mentioned the environmental organisms, I want to talk a little bit about genetically modified crops. Resistance to pests is a typical trait of genetically modified crops. How can genetically modified crops achieve this? This is because the scientists can engineer the crops to let them produce biopesticides in their plantation. In this way, when pests feed on these crops, the biopesticide can enter the gut of the pest and leading to the death of the pest. biopesticides are important for global culture. From this figure, you can see that in many countries, most of their major crops are genetically modified to produce biopesticide. For example, more than 80% of corn and cotton in US and 90% of cotton in China are genetically modified. The first generation biopesticide are Bt biopesticide. Bt biopesticide is also known as crab protein. This crab protein is originated from a microorganism called Bt. However, in recent years, we are seeing increased pest tolerance to Bt biopesticide. This is because we have planted such genetically modified crops for decades. Therefore, they keep selecting pests who have tolerance to them. To solve this problem, scientists developed the second generation biopesticide, double-strand RNA biopesticide. They are also known as RNA interference biopesticide. This is because DSI biopesticide can trigger the RNA interference in pest cell. Specifically, this DSI biopesticide can lead into the degradation of messenger RNA. As a result, the pest can no longer synthesize essential protein. DSRA biopesticides are shown to be very effective, and their effective concentration is much lower than the first generation biopesticide, Bt biopesticide. We know DSRA biopesticide can be effective in control the target pest. They may also bring potential ecological risk because we need to plant these genetically modified crops in agricultural land. And this genetically modified crop will eventually release the DSM biopesticide in the environment. This will pose potential hazards to non-target organisms, especially some pests such as the butterflies and bees, which are also insects. So we need to evaluate such biorisk. Biorisk is related to both hazard and exposure. Some biologists, they may have a better understanding in toxicity in the hazards which relate to toxicity. However, they don't have good understanding in exposure, which are related to environmental concentration and environmental fate. It is our expertise. Our environmental chemists, our environmental scientists have expertise in evaluating the environmental concentration or environmental fate. Therefore, our environmental scientists have an important role in biorisk assessment. In terms of the DSI biopesticide, the challenge is that DSI is relatively new to the environment. Therefore, the challenge is that the scientists do not have enough understanding in the environmental degradation of DSRA. So, I have two goals in my research. First, I want to develop and apply a natural method to analyze the environmental concentration of DSI biopesticide. And second, I want to elucidate processes governing the degradation of DSI in the environment. So I also want to mention that currently there are 20 to 30 reported DSI biopesticide. They are different in DSI sequences. Each of these DSI sequences, each of the DSI biopesticide have a variety of toxicity to non-target organisms. But all of these DSI biopesticide have similar initial environmental concentration and environmental fate. So my goal is to understand the ecological risk of DSR biopesticide as a whole, rather than which specific DSR biopesticide. So I choose to study the environmental concentration and environmental fate of DSR biopesticide. So in the first part of my study, my goal is to measure DSI in agricultural soil. There was a previously reported method that based on hybridization to measure DSI in soil. However, this method is not sensitive enough, and this quantification limit is higher than the estimated environmental concentration. So I intended to develop a sensitive quantification method that have a quantification limit lower than the estimated environmental concentration, which shows RT-qPCR as a tool to study the environmental concentration. This is because RT-qPCR is a sensitive quantification method because it can amplify RNA sequence during the measurement. But even with this powerful tool, we cannot just take some soil and measure DSRA use RT-qPCR directly. This is because I have some challenges during the measurement. The first challenge is that DSI in soil will readily drop to soil particles. As a result, DSI in soil samples cannot be available to RT-qPCR assay. So to better understanding the absorption process, we'll use glutide as model mineral. Glutide has a point of zero charge around 9. So at neutral pH, its surface is positive charge. As a comparison, DSI is negative charge due to the negative charge phosphate in the backbone. So because they're different charge, glutide and DSI have strong attraction. Since DSI dropped to glutide surface where phosphate group, so I propose to use phosphate species absorption comparator. So the absorption comparator, I expect them to promote attraction of DSI from glutide. So I use 3 phosphate species, orthophosphate, paraphosphate, and hexametaphosphate. You can see that all these 3 phosphate species can promote the attraction of DSI from glutide. So besides the phosphate species, I also tried the alkaline pH. This is because if the pH increase above the point of zero charge, they will reverse the surface charge of glutide and promote the attraction of DSRA. I also have a second challenge. The second challenge is that the organic matter in soil will strongly inhibit RT-qPCR. To relieve the inhibition, I can use dilution. I want to say that, so the inhibition on RT-qPCR can be seen from the increased CT value. So to relieve the inhibition, I can use dilution to decrease organic matter concentration. However, if I just use dilution, I will also greatly increase the quantification limit. So I also use a different challenge, column purification. Finally, I combine the column purification twice and then with 10 times dilution to relieve the inhibition and to keep the sensitivity of the RT-qPCR assay. So after solving the 2 challenges, I obtained a quantification method at the quantification limit much lower. You can see that much lower than estimated environmental concentration. using this quantification method, I quantified the degradation of DSRA in two soils, silty clay loam and fine sandy loam. I found that the silty clay loam absorbed more DSRA. You can see more DSRA than fine sandy loam in the initial after initial spiking. This is likely because there are differences in their surface charge. For both soil, I observed that the DSI has fast degradation within 8 hours. So compared with the previous method, this previous report method do not have low quantification limit. So they use very high initial concentration, which is 7 times higher than my initial concentration. So what will be? So will there be any problem with very high initial concentration? You can see that in their degradation kinetics, they have a lag phase in the initial stage. But in my degradation kinetics, I do not have this lag phase. So because this is likely because the RNAs in the soil may be self-recharged by the very high initial concentration. Because my initial concentration was more relevant to the environmental concentration, I expect that our result can better reflect the degradation kinetics of DSI biopesticide in the environment. So in the when I investigate the degradation of DSI in soil, I found DSI in soil can present in two forms. It can be either dissolved in the solution or can be dissolved to soil particle. So therefore, in following studies, I will have two parts: the degradation of dissolved DSI and the degradation of absorbed DSI. DSI is considered as unstable in solution at room temperature. For example, a previous study shows that DSI will dissipate in lab water and in surface water within days. The question is that why DSI is not stable? Well, we know less about DSI, the double-strand RNA. We know better about single-strand RNA. So single-strand RNA is known to have hydrolysis, which can be catalyzed by hydroxide. And this is universally present in aqueous solution. So in this reaction, the 2' hydroxyl group can be deprotonated by the hydroxide and then attack the phosphate atom, leading to the hydrolysis of the phosphor diester bond. So therefore, a representative opinion is that the RNA molecule have low chemical stability, no matter it is single-strand RNA or double-strand RNA, because single-strand RNA and double-strand RNA have the same primary structure, and they both have the phosphor diester bond. So a problem with the above opinion is that there is no direct experimental evidence. So I intended to study the stability of DSI with solid experimental evidence. And specifically, I had two research questions. First, are DSI and single-strand RNA are chemically stable under environmental relevant condition? And second, do DSI and SSI have the same alkaline hydrolysis rate? Because I intended to study the chemical stability, I need to include biofactors. So I followed the ice-free protocol. Ice is a nucleus that can degrade RNA, and it is everywhere in the environment because we human and microbes can both produce RNA. Especially for our human, we have a lot of RNA on our surfaces. This is how we protect ourselves from virus. So we use this ice-free protocol very carefully to exclude other biotic factor, so that our result only but abiotic reaction. So here I choose 4 types of RNA molecules, including single-strand RNA and double-strand RNA. I have short single-strand RNA, long single-strand RNA, short double-strand RNA, and long double-strand RNA. So I incubate these 4 types RNA at room temperature and at pH 7 for 7 months. I found that all 4 RNA molecules remain intact after incubation. They don't have degradation after 2 months of incubation. This indicates that the alkaline hydrolysis rate of both single-strand RNA and the double-strand RNA was very low at neutral pH. This result indicates that RNA-free protocol I developed is very reliable, and also it shows that I'm sure that RNA is very stable. I'm very happy for my result because today this is the longest incubation experiment of RNA incubation. So it shows that RNA degradation at neutral pH at room temperature is very stable. It's not possible for them to degrade within hours or within days. All the degradation is nothing to do with their chemical stability. So I next work on the comparison of the alkaline hydrolysis rate of SSRI and DSRI. I hypothesize that the rigidity of DSRI duplicates may hinder the requisite intramolecular attack from the cleavage of phosphate diester linkage. So to be able to measure, the alkaline hydrolysis rate, I need to raise the pH from neutral pH to very high pH. So here I increase the pH to pH 12. I found that in this pH, only SSI have hydrolysis, and DSI almost remain intact, which means that my hypothesis is correct. DSI was more stable than single-strand RNA. So my result had interesting implication for the interaction of DSI in soil, because in previous slides, you may remember that I used the high pH to promote interaction of DSI from soil, so that I was able to quantify a drop DSI. A potential concern is that the DSI have degradation during several hours interaction because of the biodegradation. So here we either the biodegradation or alkaline hydrolysis. So here we should not consider the alkaline condition, because we know that. So here we should not only consider alkaline condition, because we should also consider the RNAs from the microbes. So the blue line in the figure indicates that in clean buffers, DSI have no degradation either at neutral pH or alkaline pH, which is not surprising because we know that DSI is stable at alkaline pH. The interesting thing that in the presence, when we add soil RNAs to the system, we found that DSI will not stable at neutral pH, but can keep stable at alkaline pH. This is probably because the RNA's activity was inhibited at alkaline pH. As a comparison, the alkaline traction strategy do not work for SSI because SSI will have alkaline hydrolysis at high pH, which you can see in this figure. So at high pH, the DSI will have alkaline hydrolysis because it do not have the protection from WTE structure. So our finding is contradictory to previous suggestions that we should always operate RNA in neutral pH to avoid alkaline hydrolysis, because we can operate double-strand RNA in alkaline condition. And especially because the alkaline pH can inhibit RNA degradation, can inhibit the biodegradation. So actually, the alkaline pH can protect DSI. So after investigation on RNA stability in solution, I then studied the stability of non-formal DSI, the drop DSI. Like DNA, I have intensive absorption to soil and sediments. This is because both DNA and RNA have linear structure and they are polymers. So because they have polymers, they have multiple binding sites on their structure. The multiple binding site on DNA and RNA drive this multiple binding site can drive the absorption equilibrium to the absorption side rather than the dissolve side. So, and this strong absorption is also known as octopus effect because the multiple binding site. Theoretically, DNA in soil can be degraded by nucleus at a fast rate. However, such enzymatic degradation can be retarded if DNA is dropped to soils. As an example, DNA can be persist in soil. For example, compare with the free DSI, you can see that dropped DNA have better persistence in soils. However, it is interesting that in my previous slides, you can see that we do not see the evidence on RNA persistence in soil because we see the fast degradation of DSI in soil. So to explain this phenomenon, we hypothesize that the soil minerals may mediate the RNA degradation of DSI on their surfaces. We have such a hypothesis because the Cayley's hydrolysis have been reported for low molecular with phosphor compound on some minerals. Since RNA also have the phosphate groups in their backbone, it is possible that RNA may have similar Cayley's reaction. So again, here we use goethite and model mineral. Goethite is a mineral can be found in soils. It has a positive surface charge and neutral pH. And I have different charge because of the phosphate backbone. So here is the method I use. I firstly mix the RNA with goethite in a suspension, and then I extract the RNA from goethite by changing the chemical condition of the buffer. And finally, I centrifuge and I analyze the RNA in the supernatant. I analyze the degradation of RNA using gel electrophoresis. Therefore, I can know how much intact RNA was left in the system. So I test the stability of single-strand RNA and double-strand RNA after absorption to GOTAC. From this figure, you can see that both of the SSRI and DSRI have degradation after absorption to GOTAC, and their degradation was on time scale of hours. This time scale is relevant to their degradation kinetics in soils. In comparison, you may also remember that if there is no, if for RNA in solution, they can keep stable for months when there is no GOTAC in the system. Therefore, this result indicates that the GOTAC can catalyze the hydrolysis, catalyze the degradation of both single-strand RNA and double-strand RNA. I also determine the observed reaction. I want to prove that it is an abiotic reaction rather than biotic reaction. So I analyze their partial degradation RNA fragments. So in the gel intensity plot, you can see that the GOTAC catalyzed RNA hydrolysis have a flat and shift peaks compared with the intact RNA, which is similar to the alkaline hydrolysis. But in the figure on the right, you can see that biodegradation, they may yield several peaks. This is probably because the RNA degradation, they may have some preference on the specific sequence. So they may yield several peaks several peaks in the profile. So based on this result, I can confirm that our observed GOTAC catalyzed RNA hydrolysis is not a result of RNA degradation. It is a abiotic degradation. Besides the analysis on intact RNA, partial degraded RNA, I also analyze the final degradation product. So I analyze that GOTAC catalyzed RNA hydrolysis had the same product as alkaline hydrolysis. This means that these two reactions may have some similarities. Actually, not only test RNA, but also test DNA. You can see that in my previous result, you can see that both single-strand RNA and double-strand RNA, they both have degradation after hours. However, when I test the stability of DNA, you can see that DNA have no degradation within days. This is very interesting because that we know DNA and RNA have very similar structure, especially they have the same phosphor diester bond. So because the catalytic RNA hydrolysis may occur in the environment, I test how environmental factors will affect such reaction. I found that pH so when I change the pH of the system from neutral pH to pH 5 to lower pH, I found that there are less iron degradation. So here the column is the residual intact RNA because the TG5 have higher leftover intact RNA, so it has less degradation than the pH7, and it is same to pH9. So we expect that the lower ion degradation at pH5 is because that it cannot consistent with the lower hydroxide concentration in the solution. So this indicates that the interaction may be dependent on hydroxide concentration, and which is similar to ion alkaline hydrolysis. So similarly, we see pH9 also have higher leftover intact ion and less ion degradation. This is likely because the high pH would change the surface charge of goethite, so that absorption of ion to goethite will become loose at higher pH. Therefore, the possible hydrolysis state will become less. So besides on the pH, I also test organic matter because organic matter is everywhere in the environment. I found that if I pre-incubate the goethite with either organic matter, not only the organic matter, but also so I test not only the organic matter, but also phosphate, because they are not only organic matter in soil, but also phosphate because the use of phosphate fertilizer. So I found that if I use either organic matter or also phosphate, there will be no degradation of RNA. This means that when indicate that the organic matter and the phosphate can take up the dropship binding site on goethite, therefore they can protect RNA from degradation. So I also found that so besides goethite, we also test other minerals. For example, we also test hematite, which is another type of iron mineral. We found that both the goethite and hematite can catalyze the hydrolysis of the RNA. Is it because the leftover RNA concentration was lower than all extracted RNA? As a comparison, when we try another type of mineral, gypsite, this is a aluminum mineral, we can see that the extract RNA and the extract RNA have the same concentration as all extract RNA concentration, which means all extract RNA was not degraded. So based on this data, we found that this mineral catalyzed hydrolysis was possible for iron mineral, but not for aluminum mineral. Based on all of the data, I proposed the mechanism of goethite-mediated iron hydrolysis. In this mechanism, I first approached to the goethite surfaces due to the different charge. Then the phosphate group in RNA was coordinated by the iron atom on goethite surface. Because this is in aqueous solution, so there will be the 2' hydroxyl group can be deprotonated by the hydroxide. Both the coordination and the deprotonation facilitated, and the deprotonation can facilitate the nucleophilic attack on the phosphate atom. As a result, the phosphate diester bond will be break. This proposed mechanism is very consistent with our results. For example, I found that mineral KZ iron hydrolysis can be only occur in RNA, but only possible for RNA, but not possible for DNA. This is because the RNA have 2' hydroxyl group, but DNA does not have the 2' hydroxyl group. The involvement of 2' hydroxyl group is probably the reason why we can see this reaction for RNA, but not possible for DNA. I also expect the K-list iron hydrolysis can be very common in your environment, because you can see from the figure, you can see that iron is the second abundant metal element in soils. Therefore, expect that the K-list hydrolysis, iron mineral K-list hydrolysis, can be very common in soil. So, as a summary of my work, I studied the environmental concentration and environmental fate of DSI biopesticide, and I thinking that. So I have several summaries in my research. First, that I developed the first method that can quantify DSI in soil at concentration relevant to the environmental concentration of DSI biopesticide. And for the first time, I characterized the chemical stability of DSI and found that it is stable at neutral pH and more stable at alkaline pH than SSRI. And finally, I also found the overlooked abiotic iron hydrolysis reaction after absorption to iron oxide minerals, which are very abundant in soil. So finally, I want to say thanks to my advisor, Dr. Parker, my lab members, and funding from USDA and NSF. So now I'm happy to take your questions.
Okay, thank you, Dr. Zhang Ke. So it seems that your presentation is very interesting. So there are several questions from the audience. You mentioned that you use the soil sample to do the experiment. So the first question is that which kind of soil you used in your sample? You know, there are different types of soils.
So yeah, we use 2 types of soil. So we use 2 types of soil from our collaborator. These 2 types of soil, so one of the soil is silty clay loam and another fine sandy loam. So the silty clay loam is more like clay, it has very fine structure, and the soil particle very small. And fine sandy sample, this soil sample is more like sand. It has very large soil particle and very loose structure. We chose 2 types of different soil to represent the soil have very fine particle and another have very coarse, very large particle. So this is 2 soil. So both soil came from the real agriculture land from a farm. And we do see that because there are differences in the surface, we see the different degradation kinetics of iron in 2 soil.
Okay, thank you. So the second question is that, do you use the pH buffer to control your pH? And which kind of buffer did you use?
Yeah, so yeah, I use, so there are several places I use pH buffer to control the pH. For example, here, I can control the pH from pH 7, pH 8, pH 9, pH 10. Usually, yeah, I so I understand the question, probably understand the question that, so usually we use some buffer. We use, especially at pH 7, we usually use phosphate to control the buffer. But here, because we know that pH 7, the phosphate may have the phosphate, it can act as the dropshift comparator and may have some other effect on the traction. So here, I didn't use the phosphate as a buffer to control pH. For pH 7 and some nearby pH, I use MOPS. MOPS is a organic buffer salt. So I expect that this organic buffer salt they cannot act as the dropshift comparator. Therefore, we can know that it's only effect of pH and not because other effect caused by the phosphate buffer.
Okay, thank you. The third question is that if the iron, you present the iron, is the second abundant minerals, so why not test the aluminum in your experiment?
Yeah. Let me find the slide.
Yes.
Yeah. So my conclusion is that this K-list iron hydroxide is only possible for 2 iron mineral, but not possible for aluminum mineral. I test aluminum mineral. This is because aluminum mineral. So from this table, you can see that aluminum is the most abundant metal mineral in soil, and therefore, especially. So actually, goethite is not a non-real mineral in soil, but I can use it as a representative. So, but anyway, so aluminum is very abundant in soil. So therefore, I want to test mineral contain aluminum. Even though we found that aluminum mineral cannot catalyze iron hydrolysis, the second bond mineral, iron mineral, they can catalyze iron hydrolysis, which indicates that we can see that this reaction should be very common in soils.
Okay, so after this question, I would like to have a question from my side. So, in this PPT, we can say the less stability what happened after the RNA was absorbed to the hematite, is right?
Yeah,
so,
hematite, yeah.
Hematite. So in this interface, do you know, have you clarified the mechanism that which reaction is the driving force to degrade the RNA?
Yeah, so this is related to the mechanism. So when I propose the hydrolysis mechanism, I use goethite as a model mineral, for example, as example here. But I expect that the mechanism will be similar for hematite. This is because, so hematite and goethite have different mineral structure. They have some probably different crystal structure. But from this mechanism, you can see that this catalyzed iron hydrolysis was related to the coordination of the iron atom to the phosphate group in RNA. So no matter it is goethite or hematite, they will both have iron atom on their mineral surfaces. Therefore, even they may have some different crystal structure, as long as they have the iron atom on the mineral surfaces, they should both catalyze the hydrolysis RNA. So I expect that the hydrolysis mechanism should be similar for goethite and hematite.
Okay.
Which becomes the coordination.
Okay. So since I focus on my research in how to say, in water, in aqueous solution. So when the iron come to mind, when I say iron, the Fenton reaction always come to mind. So yes, that's why I'm so curious about these questions. Okay. So in your summary slide, you mentioned that the stability RNA at the neutral and alkaline pH. So why not acidic pH? Since there are a lot of acid rain worldwide, especially in North America. North America is one of the acid rain area. So after the acid rain, the soil is becoming acidic. Is right? So
yeah,
why you don't take this fact into consideration?
Yeah, that question makes sense to me. So first of all, I think that we test the alkaline pH, but we consider the acid pH side should have the similar result, which means that at acid pH, dsRNA should still have better stability than single-strand RNA. Because the acidic RNA also have acid chelate hydrolysis, but we expect that this hydrolysis can be also protected by the double helix structure of dsRNA. So in terms of the mechanism part, we think that both either the acid condition and the alkaline condition should yield the same result. Both dsRNA will have better result in both conditions. And second, that while we test alkaline pH when acid pH probably may be more common in the environment, the thing that although the RNA have both acid hydrolysis and the alkaline hydrolysis, actually RNA is most stable around pH 5. So as a result, the acid hydrolysis of RNA is much slower than the alkaline hydrolysis. And there is third reason. So the third reason why we test alkaline pH. So when we do this study, we are not only considering we want our conclusion to be more general, not only apply to environment but also to apply to other conditions. For example, we realized that the pest gut, so the human gut, the mammal gut, mammal stomach, they have very acid pH, but for pest, for insect, so their gut actually very alkaline pH. They have very high pH. So when we design experimental, want to test that sometimes the gut pH of the insect can reach pH 11. So when we design experiment, we also want to say that whether double-strand RNA, why double-strand RNA can be stable in the gut of the insect. So yes, our study indicated yes. Although single-strand RNA can be alkaline hydrolysis at very high pH, double-strand RNA can be very stable at very alkaline pH, which explains that why this dsRNA biopesticide can control the pest effectively.
Okay, thank you. So I have another question. Since you mentioned that in slide 16, so the challenge 2, the inhibition of organic matter on RT-qPCR, right? So it's for you. Yes, I'm very interested in the mind. Interesting idea come to my mind. You know, last year, the Nobel Prize for psychology, physiology, and medicine was awarded to archaeologist Ziwan Peber from Munich due to his development of technology to extract the genetic material from the radio of ancient human bones. So yes, when these two ideas come to me, that my question is that, is there any ideas though that you can get some help or some ideas from his technique?
Yeah, I think first of all, so why is it possible? So yeah, I also think that's interesting. So in terms of how we extract DNA from ancient bone, the first question to my mind is that why DNA can be so stable? Because we know that DNA have biodegradation. So if we just just put DNA with some microorganisms, they will degrade DNA within days. But why this DNA can be preserved in ancient bones for decades, 100 years, or some years? The reason probably because the droption. The droption bones are also minerals. So when the DNA drop to bone minerals, this droption may be one reason why they can kind of protect DNA and why they can stable in current days. That would be my understanding.
Okay, thank you. So yes, it's very interesting talk. Since I host a lot of talk, maybe you are the most one who evoke the most questions from the audience and from the host. Thank you very much. Okay.
Thank you.
Mr.
Okay. So
thank you.
Do you have any comments?
No, not comments. I think Mr. Zhang had give us a very excellent speech. I think I'm a layman of his research, but I think it's excellent work. So I have a small question. I'd like to know, just in your study, there are many kinds of biopesticides. Are there?
Yeah, I introduced 2 kinds of biopesticide. Yes.
Have you compare this reaction to different reaction on this different biocide? I'd like to know.
Okay. I got that. I got your question. So yes, DSI biopesticide is a type of biopesticide. It can be different if the RNA have different sequences. So that means that if the RNA have different sequences, so this may potentially affect their stability or degradation. But based on my data, for example, this is stability experiment at neutral pH, or I also have data on alkaline pH. So for the short and the long dsRNA, they have different sequences. And for the short ssRNA so therefore I think so, especially for dsRNA, some have short sequences, some have long sequences. I don't think the sequences affect stability because no longer about this, no matter how is the sequence of the double-stranded RNA biopesticide, they have the same phosphate diester bond. As long as the phosphate diester bond is the same, I assume that the sequences AUGC won't affect the degradation rate. That will be my understanding. So does that answer your question?
Maybe I'm not very clear, but maybe I understand it later. So
yeah.
Thank you very much. Maybe I'd sincerely invite Mr. Zhang to give us a very excellent your work and submit your paper. I sincerely invite you.
Yeah, probably in future I will consider
submit our EEH. I think your work is very excellent.
Thank you.
Okay, thank you, Dr. Zhang. So your contribution, your contribution in the future matters a lot for the EEH. Okay, so
Thank you.
So Professor Tang, do you have more comments or?
No. It's Sunday, I think, in China. There, I think many special teacher will at his deadline of National Natural Science Foundation application. It's about 10th March. This is deadline. So I think many teachers are applying this work. It's very hard.
Okay.
So maybe later we have much more conversation with you. and you can I can invite you to Nanjing. I think maybe have a chance to invite you. I think.
Sure.
Thank you very much.
Thank you.
Mr.
Okay,
thank you, Mr. He. Thank you.
Okay, you're welcome.
Professor. Bye bye.
Bye.
Okay, I would like to close this talk. So see you.
See you,
Bye.
Mr.